Hello, this is Dr. Reena giving you an input session on the introduction to microbiology. Microorganisms are the chief component of microbiology. Microbiology entails the study of microorganisms, the study of bacteria, fungi, viruses, and even eukaryotes like algae. Viruses are borderline between eukaryotes and prokaryotes. Bacteria are strictly prokaryotes. Microbiology has several disciplines. Industrial microbiology speaks about the use of fermenters in the production of antibiotics like penicillin. It speaks about the production of alcoholic beverages like wine and beer by the fermentation of grains and fruits that results in the production of CO2, carbon dioxide, and alcohol. It talks about the industrial production of enzymes like amylase enzyme produced by Bacillus amyloliquefaciens. So that is industrial microbiology. Medical microbiology, a different discipline or a branch of microbiology, speaks about the study of pathogens, virulent forms of bacteria that invade a host system, the human beings, and cause a disease. There are avirulent forms as well that are of interest. And there are mutant types that are mutated versions of virulent forms. For example, antigenic drift and antigenic variations occur on viruses, and we have new variants or new strains that are being evolved. Medical microbiology also deals with the study of viruses. Medical microbiology deals with the study of parasites, namely protozoans. That is medical microbiology. Soil microbiology, the study of microorganisms present in the soil, for example, actinomycetes like Nocardia streptomyces. Streptomyces are known to produce antibiotic streptomycin. It was Selman Waksman who discovered the antibiotic streptomycin produced by streptomyces. We have agricultural microbiology, the study of pathogens of cotton, like we have cotton bollworm, Heliothus azea, Heliothus, the bollworms are called Heliothus azea, and we have bacteria, that is entomopathogenic pesticides, bacteria used as entomopathogenic bacteria, that is Bacillus thuringiensis, Bt, that are used to be sprayed or powder that is disseminated on the crops, cotton crops, to prevent it from being attacked by Heliothus azea, etc. So that is agricultural microbiology. Also the study of several geochemical cycles like carbon cycle, nitrogen cycle is also a part and parcel of agricultural microbiology. Agricultural microbiology also deals with nitrogen fixing bacteria, Rhizobium for example. So they are known to fix atmospheric nitrogen in the root nodules of leguminous plants, of legumes. So that is agricultural microbiology. We have petroleum microbiology that deals with the studies like biosurfactants, the the oil spills that can be cleaned up by using certain pseudomonas bacteria, oil spill cleaning bacteria. So we have petroleum microbiology. We also have food microbiology that has canning industry, canned foods, and we have a major bacteria, Clostridium botulinum, that are known to cause spoilage of canned foods. Also, food microbiology speaks about dairy microbiology, where production of cheese, production of yogurt, all these are produced by the action of several fungi, and um, these are fermented products that are the result of the action of bacteria on certain substrates. So we have space microbiology, the study of microorganisms present in the planets like Mars, Jupiter, etc. Different microorganisms which are found extraterrestrial. We have space microbiology. We have water microbiology, aquatic microbiology, the study of several microorganisms which are present in the water. In water microbiology, we also study about indicator organisms that are indicators of fecal contamination of water. Fecal contaminants, as they are called. Example, Escherichia coli is an indicator organism. It indicates fecal contamination of water. In other words, sewage being dumped into water bodies, and sewage enters into the water supply, drinking water supply bodies, and this causes harmful diseases in human beings. For example, the spread of cholera. Vibrio cholerae is the pathogen that spreads through water. So we have several disciplines of microbiology, and all these disciplines deal with the study of bacteria, viruses, fungus or fungi. For example, we have Aspergillus, Penicillium, several molds. We have filamentous forms as well as the vegetative forms. So we have the unicellular yeast, unicellular fungi, which is yeast. We have molds like Penicillium, Aspergillus. We have filamentous forms like Rhizopus, Rhizopus nigricans, etc. So basically, microbiology involves a study of several bacteria, viruses that are of interest both to industries, from industries to food industries, from industries that produce antibiotics, single cell proteins or SCPs. SCPs are used as food for outer space shuttles by scientists or astronauts. So the production of SCP, single cell protein, is of value to space microbiology. So let's look at the history of microbiology. There is no microbiology without the topic spontaneous generation. There was a belief that microorganisms came out of inanimate objects like stones. It was a Greek mythology that goddess Gaia was able to give life to inanimate objects. That concept continued through several years, and we have early pioneers like John Needham, who was a die-hard advocate of spontaneous generation. He said meat, bacteria and microorganisms arose out of meat, and he said that is spontaneous generation without any presence of living component On the meat, there was germs and microorganisms that flourished and thrived and that arose. So that is spontaneous generation. Life comes into being spontaneously without a pre-existing life form. John Needham was an advocate of that. Lazzaro Spallanzani, he took meat broth or beef broth, boiled the beef broth. He conducted an experiment where the beef broth was boiled and air was prevented from entering. It was sealed. So John Needham, who was an advocate of spontaneous generation, said there was no life forms present in the beef broth because air is an important component in spontaneous generation. And that continued, that debate, spontaneous generation, abiogenesis as it is called, abiogenesis versus biogenesis. There is a biological existence of life from which microorganisms arise, or there is microorganisms that give rise to several microorganisms. Life does not originate from inanimate objects or components. So that is biogenesis. Spontaneous generation said life originated out of inanimate objects. So we spoke about John Needham, Lazzaro Spallanzani's experiments. Went down to scientists like Theodor Schwann and Francis Schulz, who conducted an experiment in which meat broth was taken and boiled, and air was treated with heat, either acid solutions or red hot flame tubes that connected into the broth or the flask that contained the broth. The tubes were heated under red hot flame, or air was passed through acid solutions and entered the flask. So air continued to exist. Air was present in this boiled meat broth, but there was no life existent in these broth solutions after several periods of incubation. All this experiment yielded reproductive results. So John Needham was still stubborn. He said the heat treatment, that is air passing through the red hot tubes into the flask that contained the broth that was being heated. He said the property of air was altered because of heat treatment and exposure to acid solutions. So again the debate continued. Spontaneous generation, sparks of that theory continued to haunt the entire development process. Then came scientists like Schroeder and von Dusch, who sealed flasks with the help of cotton plugs. So air was filtered, all the microorganisms were filtered in the cotton, and only air without the presence of microorganisms entered into the flask. And thus began the use of cotton plugs, plugging flasks, plugging test tubes in microbiology. So it was Schroeder and von Dusch who said that it was microorganisms present in the air that entered into the broth and multiplied. So the sterile broth, the broth continued to remain sterile. That means it was being heated and it continued to remain sterile only because the air was being filtered. All the microorganisms present in the air was being filtered out with the help of the cotton plug. For the one last time, Archimede Pouchet again lifted spontaneous generation and said there is spontaneous generation or abiogenesis. This time it was Louis Pasteur who brought an end to the entire debate of abiogenesis versus biogenesis, spontaneous generation versus refuting the theory of spontaneous generation, that life gave rise to life. It was not inanimate objects that gave rise to life. So Louis Pasteur took the swan neck shaped flasks and he heated the meat broth. The air that entered into the swan neck shaped flask went through a series of bends in the glass. So air, which has dust particles, and the dust particles carry microorganisms, adhered to the swan neck of the swan neck shaped flask, and air that entered into the flask that contained the heated meat broth or the beef broth was sterile air, devoid of microorganisms, devoid of dust particles in the air. So he said, Pasteur, in Sorbonne, Paris, brought an end to the entire debate. Once and for all it ended. It said, there is no spontaneous generation. Microorganisms arose from pre-existing microorganisms. Thus brought an end to the debate. So the development of microbiology went through that cycle. Then we also have scientist Louis Pasteur, who worked with the fermentation industry in Lille, France. There was good quality wine produced by good quality microorganisms in the fermented grape juice, and there was inferior quality wine that was produced by mixed culture of microorganisms and not pure culture of microorganisms. It is fermentation of grape juice that resulted in alcoholic production of wine and CO2. So Pasteur, he contributed to the economy of France by contributing and coming out with this fermentation theory, whereby he said pure cultures of microorganisms, particularly yeast, can be used to give superior quality of wine, and mixed cultures gave rise to inferior quality of wine. So Pasteur contributed to the fermentation industry, the production of wine, which was the major economy booster in France. From the problem of wine and grape juice, he went on to solve the problem of silkworm. The silk production industry was also quite flourishing in France, and pebrine was a disease of the silkworm. Pebrine was a disease of the silkworm. So the caterpillar that was infected with the protozoa, with a parasite, gave rise to pebrine, or the disease pebrine, which was the one responsible for causing silk to be not produced in its high yield, because the silkworm was infected. The caterpillar stage, which became the silkworm, was infected with the protozoa, with the parasite. So he solved the problem, and that gave rise to concepts in medical microbiology. Independently, Koch's postulate, Robert Koch came out with Koch's postulates, whereby he said that it is a germ that causes a disease. A pure culture can be cultivated from the blood of the infected animal or the patient or the man or the human being. And he said, this pure culture that is cultivated in laboratories, when injected into another animal or another human, causes the same disease. And from the blood of that animal or human being, the same isolate that first killed the animal or the human could be now isolated from this. So thus Koch's postulates contributed to the germ theory of disease. So Robert Koch independently worked in Berlin in Germany, and Pasteur worked in France. Then there was Edward Jenner, who worked with milkmaids. Milkmaids who milked the cows were never infected with smallpox because they had already cowpox virus infection in them. Cowpox was a mild virus that never really caused a disease but triggered the immune system. So most of these milkmaids who were infected with cowpox virus were resistant to smallpox viral infections. Thus, Louis Pasteur coined the word vaccination, vaxa meaning cow. He was actually honoring Edward Jenner, who worked with cowpox virus and milkmaids. So thus came or evolved vaccination and immunology. Then it was also Edward Jenner who injected James Phipps with the cowpox virus, and that boy did not get infected with smallpox. So there were independent studies done on animals. He worked with the anthrax problem, how the anthrax, Bacillus anthracis, caused anthrax, a problem of the cattle industry. So it was Bacillus anthracis that was isolated from the blood of the infected cattle. So there evolved the development of immunology. Pasteur honored Jenner by coining the term vaccination, vaxa meaning cow. So we have these great scientists contributing to the development of microbiology. Independently, soil microbiology developed. Scientist Vinogradsky was the scientist who pioneered through work of soil microorganisms. So through this session, I invite you to fall in love with microbiology, a wonderful subject, the concept of microbiology. Here we have discussed only the introduction, the different disciplines of microbiology, the history of microbiology briefly. Welcome aboard to the world of microbes. Thank you. This is Dr. Reena Nelson Antikad from Kerala, India, Asia, giving an introduction to the discipline, to the subject of microbiology. Next session we will be talking about sterilization, complete removal of microorganisms, both vegetative state and the spore state. What is vegetative state? It is a vegetative cell. Spore state is a resistant structure. So bacteria can be found in two forms: the vegetative state, the more resistant structure is a spore stage. So I just want to get you excited about the next class. It's about the complete removal. Microorganisms are ubiquitous in nature. They're found in soil, water, air. How to completely destroy microorganisms, both the spore state and the vegetative state. Then we're going to talk about disinfection. That is, inanimate object surfaces are freed of microorganisms. Disinfection is not sterilization. It is a kind or a method of partial removal of microorganisms from the inanimate surfaces. Thank you. Wait on for the next session. Thank you.